EVO2, unveiling the largest AI model for biology. Date, February 19, 2025. On February 19, 2025, a consortium of leading institutions including the Arc Institute, Stanford University, UC Berkeley, UCSF and NVIDIA unveiled EVO2, the largest AI model in the field of biology. This groundbreaking advancement represents a significant leap in the application of artificial intelligence to biological research, specifically genomics. Overview of EVO2. EVO2 was trained on a monumental dataset consisting of 128,000 genomes, which includes over 9.3 trillion nucleotides from a diverse array of life forms, ranging from humans and plants to various microorganisms. This extensive training positions EVO2 as a powerful tool capable of deciphering and manipulating genetic information with unprecedented accuracy. Key features and capabilities. One of the standout features of EVO2 is its ability to identify complex patterns within genetic sequences that would typically require years of research to uncover. The model can write entire genomes and predict the effects of genetic mutations, particularly in non-coding regions associated with diseases such as breast cancer. In evaluations, EVO2 achieved over 90% accuracy in distinguishing benign mutations from pathogenic ones in the BRCA1 gene, highlighting its potential to streamline genetic research and reduce the reliance on extensive laboratory experiments. EVO2's capabilities extend to the design of new genomes comparable in length to those found in simple bacteria. And it can process genetic sequences containing up to 1 million nucleotides simultaneously. This feature allows for the analysis of complex eukaryotic genomes, making EVO2 an invaluable resource for both understanding existing genetic structures and engineering novel biological therapies tailored to activate in specific cell types, thus minimizing unwanted side effects. Technological innovations. EVO2 builds upon its predecessor EVO1, which was focused solely on single-cell genomes. The architecture of EVO2, named StripedHyena 2, was developed to handle 30 times more data than EVO1 and can reason over 8 times as many nucleotides concurrently. This advancement is a product of collaborative efforts from AI experts including Greg Brockman, co-founder of OpenAI. Open science and collaboration. In a remarkable step towards transparency in scientific research, the developers have made EVO2's code training data and model weights fully open source. These resources are available via the Arc Institute's GitHub and integrated into NVIDIA's BioNeMo framework. This open access approach aims to foster collaboration and innovation, allowing scientists to create applications tailored to their specific biological research needs. Ethical considerations. As part of their commitment to responsible AI development, the researchers deliberately excluded human pathogens from EVO2's training data, addressing potential ethical concerns associated with the deployment of large-scale biological AI. This precaution reflects a proactive stance toward the responsible use of AI in sensitive areas of health and biology. Conclusion. EVO2 marks a pivotal moment in the field of generative biology, enabling machines to read, write, and think in the language of nucleotides. Patrick Su, a key developer of the model, expressed enthusiasm about the future applications that the research community will derive from this foundational model. As scientists delve into EVO2's capabilities, its potential to tackle critical health and disease challenges is expected to grow, establishing it as an essential asset in the evolution of biological research. Reception and future directions. The announcement has garnered attention across the scientific community, with researchers praising the innovative engineering behind EVO2. The initial reception indicates a strong interest in its applications, with anticipation for independent benchmarks to validate its performance. In addition, the EVO2 model is viewed as a platform upon which specialized AI tools for biology can be built, akin to an operating system kernel that supports a variety of applications. The team envisions continued refinement and application of EVO2, hinting at an exciting future where AI significantly contributes to our understanding of biological systems and the development of novel therapeutic strategies. Significance in the field. The introduction of EVO2 not only underscores the rapid advancements in AI and machine learning but also highlights the collaborative spirit of the institutions involved. By pooling resources, expertise, and data, the research community is setting a precedent for future endeavors in the ever-evolving landscape of biological research. As EVO2 is explored further, its role in decoding complex genetic information and aiding in the design of innovative biological therapies will likely position it as a cornerstone of modern genomics and synthetic biology, addressing some of humanity's most pressing health challenges. Sources: 1. Nature article on EVO2. 2. HP Choir coverage of EVO2. 3. UC Berkeley's report on EVO2. Overall, EVO2 represents a monumental achievement in the intersection of artificial intelligence and biology, promising to unlock new avenues for research and therapeutic development in the years to come. EVO2, unveiled on February 19, 2025, surfaces as a pivotal advancement in the field of artificial intelligence applied to biological research, boasting the title of the largest AI model for biology to date. Developed through collaborative efforts from the Arc Institute, Stanford University, UC Berkeley, UCSF, and NVIDIA, this model is unprecedented in scale and capability, having been trained on an immense dataset of 128,000 genomes, which collectively encompass over 9.3 trillion nucleotides from an extensive array of lifeforms, including humans, plants, and various microorganisms. Key features and capabilities. EVO2's design allows it to identify intricate patterns in genetic sequences, significantly accelerating the discovery process for researchers. The model can generate entire genomes and predict the implications of genetic mutations, with particular emphasis on non-coding regions associated with diseases such as breast cancer. Notably, EVO2 achieved over 90% accuracy in distinguishing between benign and pathogenic mutations in the BRCA1 gene, illustrating its capacity to enhance genetic research and potentially reduce the necessity for extensive laboratory investigations. Additionally, EVO2 can design new genomes mirroring the length of simple bacterial genomes and process genetic sequences with lengths extending up to 1 million nucleotides at once. This makes it adept at analyzing complex eukaryotic genomes, positioning EVO2 as an essential tool for both understanding existing genomic structures and engineering novel biological therapies that can activate specifically within targeted cell types to minimize adverse effects. Technological innovations. EVO2's architecture, StripedHyena 2, marks a substantial evolution from its predecessor EVO1, which was limited to single-cell genomes. With enhancements allowing it to manage 30 times more data compared to EVO1, this new architecture facilitates reasoning over 8 times as many nucleotides simultaneously, significantly expanding the model's analytical capabilities. Open science and collaboration. A noteworthy aspect of EVO2's development is its commitment to open science. The model's code, training data, and weights are fully accessible to the public via the Arc Institute's GitHub repository and integrated into NVIDIA's BioNeMo framework. This open access approach aims to foster collaboration and innovation, inviting scientists to develop applications tailored to their specific research needs within the biological domain. Ethical considerations. In the realm of ethical development, the researchers took proactive measures by excluding human pathogens from EVO2's training data. This decision reflects a conscious effort to address potential ethical concerns associated with the deployment of large-scale biological AI, emphasizing responsible research practices. Conclusion. EVO2 heralds a transformative era in generative biology, enabling machines to read, write, and think in the language of nucleotides. Patrick Hsu, a leading figure in its development, expresses optimism regarding the future applications that the research community will derive from this foundational model. As scientists explore EVO2's capabilities, its role in tackling critical health and disease challenges is poised to expand, solidifying its status as a vital asset in the ongoing evolution of biological research. While the overarching narrative of EVO2 emphasizes its scale, capabilities, and collaborative nature, some counterintuitive aspects are worth noting. 1. Open source versus competition. The decision to make Evo2 open source may seem counterintuitive for proprietary advancements in AI, where companies typically guard their models for competitive advantage. However, this approach fosters a collaborative environment that could accelerate innovation, ultimately leading to breakthroughs that may benefit all stakeholders involved. 2. Focus on non-coding regions. Traditionally, genetic research has heavily favored coding regions, as they directly correspond to protein synthesis. Evo2's emphasis on non-coding regions areas often overlooked challenges the prevailing focus, suggesting that significant discoveries regarding gene regulation and disease may lie in these expansive but misunderstood regions of the genome. 3. Ethical exclusion of pathogens. While the exclusion of human pathogens from the training data is a responsible choice, it could also be seen as limiting the model's applicability in crucial areas of infectious disease research. The decision reflects a balancing act between ethical AI development and the potential for broad application in understanding pathogenic mechanisms. 4. Generalist versus specialist models. Evo2's characterization as a generalist model might lead some to question its efficacy in specialized tasks. However, its ability to generalize across diverse biological data suggests that it can serve as a robust foundation for developing specialized models tailored for specific research applications, potentially leading to unexpected efficiencies and insights. 5. Immediacy of applications. While the immediate applications of Evo2, such as identifying mutations and designing genomes, are promising, the long-term impact on fields like synthetic biology and personalized medicine may unfold in unpredictable ways. The model's release could catalyze a broader rethinking of biological design and engineering, transcending current methodologies. In summary, Evo2 stands at the forefront of a new era in biological research, equipped with capabilities that challenge existing paradigms and open doors to previously unimaginable advancements. Its development, characterized by collaboration, transparency, and ethical considerations, signifies a significant leap forward in the integration of AI with the life sciences. The unveiling of Evo2, the largest AI model in biology, marks a significant leap forward in the intersection of artificial intelligence and biological research. While many reports focus on its size, capabilities, and collaborative development, several nuanced aspects are often overlooked that carry substantial implications for the future of biological research in healthcare. 1. Open source commitment and its implications. Evo2's full open-source release, including its code, training data, and model weights, represents a landmark shift towards transparency in scientific research. This move is significant because it democratizes access to a powerful tool that can be adapted for diverse applications in biology. By making this model openly available, researchers worldwide can build upon it, fostering innovation and collaboration across institutions that may not have the resources to develop such models independently. The potential for community-driven advancements is immense. This collaborative environment can accelerate discoveries as researchers can modify and refine Evo2 to suit specific needs in their studies, ranging from gene editing to synthetic biology. The impact of this open-source approach could lead to breakthroughs that might not have been possible within a more closed framework, emphasizing the model's role as a foundation for future research rather than a standalone product. 2. Handling of non-coding regions and disease implications. While Evo2 is celebrated for its ability to process both coding and non-coding regions of DNA, the emphasis on its proficiency in interpreting non-coding mutations is particularly important. Non-coding regions, which previously received less attention, have been increasingly implicated in various diseases, including cancer. Evo2's demonstrated accuracy in predicting the pathogenicity of non-coding mutations, especially in genes like BRCA1, may revolutionize how genetic testing and counseling are approached. The ability to accurately identify benign versus pathogenic mutations could streamline genetic research and clinical diagnostics, reducing the need for extensive laboratory work and potentially leading to earlier interventions in disease processes. This capability is particularly critical given the growing recognition of the role non-coding DNA plays in health and disease, suggesting that EVO2 might facilitate a deeper understanding of genetic risk factors and personalized medicine. 3. Ethical considerations in model development. The exclusion of human pathogens from EVO2's training data is a significant ethical consideration that may be overlooked. By consciously avoiding pathogens, the developers aim to mitigate risks associated with the misuse of AI in biological research, such as the potential development of harmful gene editing tools. This decision underscores a proactive approach to responsible AI development, addressing concerns that could arise from powerful models capable of manipulating genetic information. This ethical stance is crucial as AI's role in biology expands. It raises essential discussions about the responsibilities of researchers and developers in ensuring that their technologies are used for the benefit of society and not for harmful purposes. By establishing these ethical guidelines, the creators of EVO2 contribute to a framework that prioritizes safety and responsibility, which is vital in a field as sensitive as genetic research. 4. Architectural innovations in computational power. The advancements in AI architecture that EVO2 represents, particularly the StripedHyena 2 framework, enable the model to process vast amounts of genetic data with unprecedented efficiency. The ability to analyze sequences containing up to 1 million nucleotides simultaneously is a significant technical achievement that not only enhances the model's performance but also sets a precedent for future AI developments in biology. This shift in architecture allows EVO2 to handle the complexities of eukaryotic genomes, which are inherently more intricate than prokaryotic ones. The implications of this capability extend beyond mere data processing. They pave the way for more sophisticated analyses and applications in synthetic biology, such as the design of new genomes and tailored biological therapies. The potential to create targeted therapies that activate in specific cell types could revolutionize treatment approaches, reducing side effects and improving patient outcomes. 5. Generative biology as a new field. EVO2 is not just an AI model, it signifies the emergence of generative biology as a distinct field. The ability of machines to read, write, and think in the language of nucleotides introduces a new paradigm in biological research where computational models can generate biological insights and innovations. This generative aspect could lead to novel applications in various domains, including drug discovery, environmental biology, and agricultural biotechnology. The recognition of generative biology as a new frontier emphasizes the need for interdisciplinary collaboration among computer scientists, biologists, and ethicists. As this field evolves, it will challenge existing methodologies and inspire new research questions, highlighting the importance of integrating AI into biological sciences comprehensively. Conclusion. EVO2 represents a watershed moment in the application of AI to biology with far-reaching implications. Its open-source nature fosters collaboration and innovation, while its capabilities in interpreting complex genetic data, particularly in non-coding regions, could transform genetic research and clinical practice. The ethical considerations surrounding its development and deployment establish a responsible framework for future AI advancements in biology. Finally, as generative biology emerges as a new field, the potential for interdisciplinary collaboration promises to unlock unprecedented opportunities in understanding and manipulating the biological world. These aspects, often overshadowed by the model's sheer scale and technical achievements, are crucial to understanding its significance in the scientific landscape. 4 valuable points for in-depth analysis and discussion on EVO2. 1. Scale and scope of data utilization. EVO2 stands out as the largest AI model in biology, trained on an unprecedented dataset of 128,000 genomes encompassing 9.3 trillion nucleotides from a diverse range of organisms, including humans, plants, and microorganisms. This immense scale not only surpasses its predecessor EVO1, but also allows EVO2 to identify complex patterns in genetic sequences that would take researchers years to uncover. By incorporating a wide array of genomes from both prokaryotic and eukaryotic lifeforms, EVO2 is positioned to unlock insights across various biological domains. The implications of this extensive dataset are profound as they enable researchers to analyze genetic variations and disease-causing mutations with unprecedented accuracy and efficiency. The ability to process genetic sequences of up to 1 million nucleotides at once further enhances the model's capacity to interpret complex eukaryotic genomes, making it a powerful tool for advancing genetic research and therapeutic design. 2. Technological innovations and architectural advancements. The architectural advancements of EVO2, particularly the StripedHyena 2 framework, mark a significant leap in AI design for biological applications. By enabling the model to handle 30 times more data than EVO1 and reason over 8 times as many nucleotides simultaneously. This new architecture is crucial for analyzing long and complex genetic sequences. The collaboration with notable AI figures including Greg Brockman from OpenAI underscores the importance of interdisciplinary efforts in pushing the boundaries of AI capabilities. This innovation not only enhances the model's performance but also sets a precedent for future AI applications in biology, suggesting that tailored architectures can lead to breakthroughs in understanding biological systems and processes. 3. Open science and collaborative research. EVO2's commitment to open science is a cornerstone of its design and development. By making the model's code, training data, and weights publicly available via platforms like GitHub and NVIDIA's BioNeMo framework, the creators aim to foster collaboration and innovation within the scientific community. This open access approach allows researchers worldwide to leverage EVO2's capabilities for various applications, effectively creating an app store for biology. The emphasis on transparency and interpretability, including tools developed to visualize how the model identifies biological patterns, contributes to responsible research practices and encourages user engagement in refining and applying the model. The collaborative nature of the project, involving institutions like Stanford University, UC Berkeley, and UCSF, exemplifies how interdisciplinary partnerships can drive significant advancements in scientific research. 4. Ethical considerations and responsible AI development. The researchers behind EVO2 have taken noteworthy steps to address ethical concerns associated with large-scale AI models in biology. By consciously excluding human pathogens from the training data, they aim to mitigate risks related to misuse or unintended consequences of the model's deployment. This ethical framework is essential in ensuring that the technology is applied responsibly and does not contribute to harmful biological research or applications. As AI continues to permeate various fields, the emphasis on ethical considerations in model development and deployment will be crucial in guiding the responsible use of AI technologies in sensitive areas like genomics and health research. Engaging with the ethical implications of AI in biology not only promotes responsible scientific practices but also builds public trust in the research community's efforts to harness AI for beneficial outcomes. These 4 points encapsulate the transformative potential of EVO2 in biological research, highlighting its scale, technological advancements, collaborative nature, and ethical considerations. The ongoing exploration of EVO2's capabilities will likely lead to significant contributions in understanding genetics, improving health outcomes, and advancing the field of generative biology. 1. The Human Genome Project 1990–2003. This landmark scientific endeavor aimed to map all the genes in the human genome. Involving an international collaboration of scientists, the project successfully identified and sequenced all 3 billion DNA base pairs in the human genome, which has had profound implications for genetics, medicine, and biotechnology. The completion of the Human Genome Project has paved the way for advancements in personalized medicine, genetic disorders understanding, and the development of new treatments. 2. CRISPR-Cas9 gene editing technology 2012–present. The development of CRISPR-Cas9 has revolutionized genetic engineering since its introduction. This technology utilizes a natural defense mechanism in bacteria to edit genes with unprecedented precision and efficiency. Researchers can target specific DNA sequences for modification, allowing for potential treatments of genetic disorders, advancements in agricultural biotechnology, and significant breakthroughs in biological research. The ethical implications and potential applications of CRISPR continue to be a major topic of discussion in the scientific community. 3. IBM Watson for Oncology 2011 present. IBM Watson for Oncology represents an attempt to harness artificial intelligence to improve cancer treatment. Trained on vast amounts of medical literature and clinical data, Watson assists oncologists by providing evidence-based treatment recommendations tailored to individual patient cases. Although it has faced challenges in clinical implementation and skepticism about its effectiveness, Watson's development reflects the growing intersection of AI and healthcare, similar to the aspirations surrounding EVO2 in enhancing biological research and therapeutic design. Future outlook for EVO2, predictions for biological AI. EVO2, launched on February 19, 2025, marks a significant milestone in the intersection of artificial intelligence AI and biological research, representing the largest AI model in biology to date. Its vast dataset, encompassing 128,000 genomes and 9.3 trillion nucleotides, positions it as a transformative tool for genetic research and synthetic biology. As we look to the future, several key predictions emerge regarding the impact of EVO2 on the fields of biology, medicine, and AI technology. 1. Accelerated genetic research. EVO2's capability to identify complex patterns in genetic sequences will likely revolutionize genetic research. Predictions suggest that researchers will leverage EVO2 to uncover insights into genetic diseases at unprecedented speeds. The ability to accurately predict the effects of genetic mutations, especially in non-coding regions linked to diseases like breast cancer, will streamline research processes and significantly reduce the time and resources required for laboratory experiments. This acceleration could lead to faster identification of genetic markers for various diseases, potentially improving early diagnosis and treatment strategies. 2. Advances in synthetic biology. As EVO2 can design new genomes, it will likely play a pivotal role in the field of synthetic biology. Researchers may utilize EVO2 to create tailored genomes for specific applications, such as engineering microorganisms for bioremediation or developing crops with enhanced resilience to environmental stressors. The model's ability to generate genetic codes that activate in targeted cell types could lead to breakthroughs in gene therapy, where precision medicine becomes the norm rather than the exception. These advancements may result in more effective treatments with minimized side effects, ushering in a new era of personalized medicine. 3. Collaborative research ecosystem. The open-source nature of EVO2, with its code training data and model weights available to the research community, is expected to foster a collaborative ecosystem. Scientists across various disciplines will likely build upon EVO2 to develop specialized AI models tailored to specific biological challenges. This collaboration could result in a proliferation of innovative applications akin to an app store for biology. As researchers create tools and solutions that leverage EVO2's foundational capabilities, the outcome may be a robust community of interdisciplinary researchers addressing complex biological questions collaboratively. 4. Ethical framework and responsible AI use. With the deployment of powerful AI models like EVO2, ethical considerations will remain a critical focus. The researchers behind EVO2 have already taken steps to address potential ethical concerns by excluding human pathogens from the training data. Moving forward, we can expect the establishment of more comprehensive ethical frameworks governing the use of biological AI. This may include guidelines for responsible AI deployment, ensuring that advancements do not come at the cost of safety or ethical integrity. The dialogue surrounding the ethical implications of AI in biology will likely expand, involving stakeholders from various sectors, including policymakers, ethicists, and the scientific community. 5. Integration with other emerging technologies. The application of EVO2 will likely extend beyond biology into other domains including computational biology, bioinformatics, and even environmental science. The integration of EVO2 with other emerging technologies, such as CRISPR gene editing, robotics, and machine learning, could lead to innovative solutions for pressing global challenges including climate change, food security, and disease outbreaks. For instance, combining EVO2's predictive capabilities with CRISPR technology could facilitate the rapid development of gene editing strategies aimed at combating genetic disorders and enhancing agricultural productivity. 6. Enhanced understanding of evolutionary biology. EVO2's capacity to analyze and predict genetic patterns across diverse life forms will contribute significantly to the field of evolutionary biology. It may provide insights into evolutionary processes, helping researchers understand how genetic variations influence adaptability and survival in changing environments. This enhanced understanding could lead to new theories in evolutionary science, influencing both academic research and practical applications in conservation biology and biodiversity preservation. 7. Health and disease management. The implications of EVO2 for healthcare are profound. As it aids in identifying disease-causing mutations and designing precise treatments, EVO2 could facilitate the development of new diagnostic tools and therapeutic strategies. The healthcare industry may see a shift towards more data-driven approaches, with AI playing a fundamental role in patient management, from personalized treatment plans to predictive analytics for disease outbreaks. This could ultimately lead to improved public health outcomes and a more proactive approach to disease prevention. Conclusion. The future outlook for EVO2 is characterized by a landscape of opportunities and challenges that will shape the future of biological research in medicine. As we harness the power of AI to explore the complexities of life, EVO2 stands poised to be a cornerstone in this evolving journey, driving innovation, fostering collaboration, and addressing some of humanity's most pressing health and environmental challenges. The journey ahead will require careful navigation of ethical considerations and a commitment to responsible use, ensuring that the benefits of such technology are realized sustainably and equitably.